Hi everyone. Tonight I'm going to be talking about human and animal subjects, other things that are regulated, conflict of interest, and whistleblowers. So that sounds like a bunch of topics, but really I think this is kind of like my lawyer. I'm going to be a legal advisor tonight, but these are all things that have laws and regulations around them. And then conflict of interest is what to do if people may break some of these rules, and whistleblowers will tell you about what to do if you don't see compliance. So I feel like this isn't an end-all be-all kind of introduction to any of these, but one of the things I've realized is that a lot of times chemists don't even know what's regulated. And so my goal here is to give you a quick overview of what's regulated so that later on, if you run into any of this in your research, you'll at least know, oh, that's a hot topic. I got to go and look that up and figure out what I need to do to be able to do that legally. And it's really important that you do these things legally, because if not, like, these are the kinds of things that you can get sued for, you could get kicked out of science for, not be able to get grants, not be able to publish your papers, etc., etc. They're really serious because people think that these are the kinds of things that we need to make sure that we're regulating. Okay. So the first one we're going to cover tonight is human research. So obviously human research is any research that involves a human or data about a human. We'll talk about that on the next slide. So, examples of things might be working with tissue or blood samples. Chemists do that a lot. You might not actually ever see the human, but you work with a sample from it. Giving an experimental drug or treatment. Maybe you're in the drug discovery field, and that's, what you hope to do someday. But it also includes health data from medical records. Any data about kind of outcomes or that kind of thing, as well as any survey or behavioral data. And so the number one thing you need to remember is that to be able to do anything that's related to a human, you must put a protocol in and be approved by the Institutional Review Board, or IRB. You're going to see 1,000 acronyms tonight. Everything has a board, basically, and everything needs approval. And UVA actually has several different IRBs. They have some that work on the more medical side, some that work on the more behavioral and survey side. So there's different expertise of different IRBs, but you have to put that IRB in before you start any research, right? You can't go ahead and do the research and then later go, oh, I want to publish this now and I never got approval. I'm going to go back and get that. That's not how it works. You're not going to be able to publish that data that you acquired without getting approval. Then, once you get approval, one of the things that you have to do is go through a bunch of human subjects research training, right? Research training in HIPAA rules, which are privacy, research training in a bunch of best practices, keeping everybody safe. And so, there's training to go through, frankly, for all of these things. So this is the NIH again, and some of their rules for human subjects research. So they define it as research involving a living individual about whom data or biospecimens are obtained, used, studied, analyzed through interaction, intervention, or identifiable private information is used, studied, analyzed, or generated. So there's a few key things there. Living, I guess if you're dead, they don't care. Interactions, interventions, right? Private information. They're really concerned about privacy right at the moment. So again, they have some examples of human subject research that you might do. And again, a lot of them of course, I'm giving an experimental drug, that's a human subject research, but even interviewing somebody can be that, administering a psychological test, that kind of thing, or testing a new educational research technique. So if you're submitting an NIH application, you have to and you're going to use human subjects or tissue or anything like that, you have to put in a protection of human subjects attachment, and then if kind of thing that your institution is protecting human welfare and that kind of thing. Now, there are a few exemptions to this, and so there are 8 categories of exemptions shown here. I'm not going to go through all of them. Normal educational practices are exempt, but the one that we often see in kind of chemistry is this E4, exemption 4. And that is the collection or study of data or specimens if publicly available or recorded such that they can't be identified. So we often call this de-identified samples, right? Like somewhere took some tissue, you want to look for a protein in that tissue, right? But you have no idea who those people are. If the samples are de-identified, and particularly if they already exist at the time of your study, sometimes you can get an exemption from human subjects because of that. But it's a little bit tricky because let's say you want to study, proteins in a disease in a certain tissue, and your collaborator down at the med school is going to collect those tissues and then give them to you. Is that de-identified research? No, because the collaborator and you are directly involved in the same project, right? And you're collecting new samples. So this de-identified research sample is not an out for, well, I didn't meet the patient, so I'm okay. No, it's the whole project. And so if somebody in the project has met the patient, it's not de-identified. Usually the data had to exist before you started. So there's a lot of sticky points here. And again, we're not going to cover them all. If you're doing human subjects research, you should have a lot more training, again, through the university and with your PI. But I want you to know that there are some sticky points. Sometimes there are protocols that ask lab members to give samples for research. Because sometimes people just want to, detect something. They're developing a test in blood or urine or that kind of thing. And, well, where are they going to get blood and urine on a regular basis? Oh, well, a lab member can give it. I want to remind you that you can never be compelled to give any subject. If you don't feel comfortable giving it, you don't have to, and your performance should never be based on it. So there are times when people do this, but it's a little bit sticky, and you should never feel like you have to. The other thing is that sometimes, people are excited about doing education research, like in chemistry education, like I'm going to study, my students to see if they do better if I teach in a certain way. And so using your own students to do education research is really a sticky point. And a lot of people who aren't education researchers, but just good teachers, want to do it, and they don't know all the rules. So it can be done, but you have to make sure there's no penalty if the student chooses not to participate, right? And again, it's best to hook up with somebody in the educational school or talk to the IRB about the best practices. Our Center for Teaching and Learning actually can give seminars on the best practices here. So it can be done, but you should know that you can't just automatically assume anything that you collect in class can be given. And again, reminder that when you're working with students, hopefully you learned this in TA training, there are all the HIPAA regulations. Sorry, not HIPAA, FERPA. There's too many acronyms. And that's the educational privacy, right? So you can never put students' answers, with any identifiable information or anything identifiable about a student. Don't even take pictures of a Zoom meeting, right, and put them up with students. They have a lot of privacy rights. Okay, and then the last one is the thing that I talked about with de-identified samples, that theoretically you're exempt if they already exist, but if your collaborator collects them, and you're again, in the same project, the IRB is probably still needed. They used to have a rule, and I'm not sure if it's exactly the same, but when I was on study section, that if you wanted to be exempt for de-identified samples, the samples actually had to already exist before the grant started. So if they were going to be collected at all during the time of the grant, you had to write the IRB, and you had to get IRB approval. So it's a really narrow exemption, and you shouldn't just think, oh, of course my stuff is E4 exempt. And how do you know that it's E4 exempt? You send it to the IRB, and they tell you it's exempt. So it's not even like you get out of it necessarily, that you have to prove that it's exempt to the NIH or to your IRB. Okay, so that's human subjects, and quite frankly, animals somewhat mirror human subjects, right? There was a Laboratory Animal Welfare Act that was passed by Congress in 1966, and this covers things like cats, dogs, non-human primates, guinea pigs, and rabbits. And so if you use any of these types of animals, they're regulated by the USDA, meaning the USDA can actually come into UVA and look at our facilities and make sure we're treating these species properly. What it does not include are mice, rats, and birds. There's a push a few years ago to try and get it to include that, and the animal researchers really pushed back. So the USDA does not come in to look and see if we're treating our mice, rats, or birds correctly. However, there's a public health service policy on human care and the use of laboratory animals, and that covers all vertebrate animals, such as mice, rats, and fish. So there, we go for most of the things that people typically use in the laboratory, like mice and rats. And so you have to follow these regulations for federal funding, and there's accreditation, again, by another huge acronym called ALAC. So basically, once every 3 years, we get this team of people who descend on the university for 3 or 4 days to accredit us for animal research. And so this is required for anybody to be able to get a federal grant to work on animals. So we have to be accredited. Okay, So what do you want to do if you want to go to work on animals? Again, there's a review board. In this case, it's called the Institutional Animal Care and Use Committee, which is also called the IACUC. They did not pick IRB as better, but it's basically the equivalent of the IRB. And again, anytime you want to do anything to an animal, a vertebrate animal, you must get approval before you begin the research. That's the most important part. If you don't remember anything else, you must always get approval before you start. It typically applies for any vertebrate animals, but it doesn't apply for things that aren't vertebrates like insects, C. elegans, or things like cell cultures that are immortalized. So there's, not everything requires it. And then what are they looking for? There's, whenever we fill these out, there's like 90 questions to answer. But there's a real focus on animal welfare, right? Minimizing pain and distress, doing whatever we can to minimize the number of animals and to minimize their pain and distress. And similar to the humans, if you're doing animal research, then you must go through university training, occupational health surveillance, as well to keep the researchers healthy. So again, people can have allergies or sometimes even get diseases from animals. So they have an occupational health program to make sure that you're okay. So that's animals. The next thing that's regulated that you need to know about is biosafety. So the biohazards are regulated by the University of Virginia Environmental Health and Safety Institutional Biosafety Committee, the IBC. And again, what's a biohazard? Here's just a few of them that are listed on their website. Adeno-associated viral vectors. These have become really popular to put in different genes. Some of you are working on like fluorescent sensors, and you might use these viral vectors. Things like biotoxins, my lab uses biotoxins, are regulated. Human-derived materials, human gene transfer, again, if you're trying to do some sort of genetic manipulation, and recombinant and synthetic nucleic acid molecules, and then what they call select agents, which are somewhat like biotoxins, but things that kill you. If you use it wrong, those select agents can kill you, and so they're regulated by the government. They don't want those getting out to the general population. And again, any research protocol using any of these types of things must be approved before beginning on the work. And then again, it's under environmental health and safety, so the safety people come out and do inspections to make sure you're using the right kind of safety things, right? So for biosafety, there's often biosafety hoods that have to be used, right? There's different BSL levels for biosafety, 1, 2, 3, and 4, and so they're going to make sure that you're using all the proper protocols and safety and PPE and all of that to make sure that you're working on that. They also, Environmental Safety, Health and Safety also has a radiation safety committee, and they regulate both radioactive tracers, like radioactive materials, as well as radio-labeled drugs. And again, they always you have to write a protocol beforehand, and then they'll actively inspect and monitor radiation levels, give people like little badges to wear, look at the rooms, et cetera, for radiation safety. I don't know that we have a lot of people using radioactive things in chemistry, but we certainly can, right? I mean, we chemists know how to make radioactive molecules. So again, I want you to have it on your radar screen that radiation is something that's regulated by the university. And again, they're also following OSHA regulations to make sure that your health and safety, as a worker, are being maintained. Okay, next is drugs and controlled substances. And so these are regulated more by the federal government, the Drug Enforcement Agency. And so the PI to have these must have a federal license. So example of some controlled substances include cocaine, ecstasy, heroin, and ketamine. My lab owns 3 out of the 4 of those. And so yes, I have a drug license. But there's a new kind of regulation that's gotten a lot of PIs now to have a state drug license. And that's that anything that we require a prescription is also regulated by the state. And that means the PI has to get a state license from the Board of Pharmacy. This is a relatively new requirement. We used to be able to just use the kind of like the license of the veterinarian who is over in charge of animals, and they won't let you do that anymore. So again, many times people are going to give a drug like, you said ketamine, that's special K. It can be abused, but it's also a really common sedative that we give to animals to do surgeries. All right, so a lot of people own almost all the anesthetics that we use for anything in animals are prescription. And so many of your PIs will have these state licenses. They've got to be renewed yearly, and the labs are then inspected by the government. I get with the federal and the state government come out and inspect my lab on a regular basis. What are the rules? What are they looking for? All of my drugs must be properly secured. They must be in locked boxes, et cetera, and all of the use must be documented in logs. This goes for biosafety too. Again, my lab, I feel like my lab has just about all of these. We don't have radiation service, safety, but I have been human certified, animal certified, biosafety certified, drug certified. Man, I know how to do all of these. It's just a lot of rigmarole, but you can get it done. But everything has to be logged, right? Anytime you use a biotoxin or anytime you, use a controlled substance, you got to log it in, you got to log it out. So you can tell that nothing's being stolen or given out to the public. Oops, that wasn't supposed to be there. We'll get back to that. Okay, financial conflict of interest. Okay, so now I've switched from those are all the things that were regulated. Financial, now we're going to go to conflict of interest. And again, this kind of falls under the same vein of things you must report to the university and make sure that you manage well. And again, I think you should know about these, even though grad students aren't likely to have a lot of conflict of interest, but you might start working with PIs that do, and you should just know about the rules in science as you go up the ladder, right? That somebody should have trained you on, like, what is a conflict of interest? So we'll start with the one that's most obvious, and that's financial conflict of interest. That's what an investigator has a significant financial interest that could directly or significantly affect the design, conduct, or reporting of the funded research. So the way this is managed, even though NIH and like maybe NSF or other people care about this, they kind of let the institution manage it. So all financial conflicts of interest must be reported to the institution. I have to fill out a form every year about my conflicts of interest and any place I get paid from, any other sources of income, that kind of thing. And then if the institution deems that I have an actual conflict of interest in something that I'm doing, then they come up with some sort of management plan to make sure that it's not going to affect my work. So there's a real problem here, and that is there's often a blurring of the lines between a company and university research. So in some ways, the university is encouraging everybody, start your own company, right? Be an entrepreneur. But when you do that, then you have to keep the interests separate. There was a story a couple years back where a professor actually got arrested for using university property and university resources for their company. So this is serious business, and the lines are really blurry. What's collaborative research, that can be done at the university, what should only be done at the company, et cetera, et cetera. Just so again, the university has a very liberal policy, in my opinion, for professors. We are allowed to be a consultant up to 1 day a week, which means I can get paid to do other research, other work up to 1 day a week. And so it is possible that, your advisor could spend some time at a company and it could be okay. I want you to know that they can spend up to 1/5 of their time at the company, and that would be okay with the university and their job things. But there's a real need to kind of keep things separate, that their students aren't always doing work for the company, et cetera, et cetera. And it's kind of a sticky point. But if you ever have any problem, you can come talk to me or somebody else in the university. I'll tell you who the main person in the university is that you can talk to if things aren't separate. And then if you do have a conflict of interest, like I started this company and they gave money, or I tested a drug for the drug manufacturer or something like that, it needs to be disclosed both whenever you give a talk or in a publication. Quite frankly, when I'm down at the med school, I often see, if an investigator has started a company, they'll just put it on their first slide. I have a conflict of interest. I started this company, right? And they're now like developing whatever. I haven't seen it as much at chemistry seminar, but you'll see it. Okay, there's also another type of conflict of interest for peer review. And that is, should you review somebody's grant? And so generally, you should not review papers or grants from family members. That's pretty obvious. Close personal friends or research mentors. Okay, what's a close personal friend? Many people become friendly in science. Like I see the same people. They're, at conferences all the time. We go out to dinner, we go out to lunch. We really enjoy, being with each other, that's not a close personal friend, actually. The one definition I've seen somebody use is that, would you invite them to your wedding? That's kind of like the definition of close personal friend. And so the reality is that most of the time, you can be friends in science and not call it their close personal friend. That we all kind of have networks, and we're going to assume that we can kind of push past that to be friends and still review things fairly. NSF has a policy that they won't let you review a grant with anyone you've published it in the last 5 years. They make you list every single author that you've published with. So that's kind of how they view collaborators. So let's look at the NIH thing. So you can't even be on the study section is basically the grant review panel. You can't even be on it if you're on an application in a major role, right? Obviously, you wouldn't be wanting to review other people's if that panel is going to review yours. If you're a member of an advisory committee, or if you had a direct financial benefit. So you can't even be anywhere near the review process. Now, sometimes you serve on the review panel, but then you go out of the room. You don't actually participate in the review of certain applications. That's the middle one. So there's institutional. So like if I'm on a grant review and there's somebody else from the University of Virginia, then I'm automatically in conflict and I can't review their application. So I've been on study sections before where like Don Hunt's proposals were there, or Rebecca Pompano's, right? There are other analytical faculty, and I have to leave the room and I don't know what happens. So again, they don't want you to be able to influence that. A collaborator within the last 3 years, or another professional relationship, such as a mentor, that kind of thing. Again, sometimes you'll give a letter of support, that you'll help somebody for something. Obviously, if you're giving a letter of support, you shouldn't be reviewing that application. If you're on our advisory board, sometimes there's big projects, right? Like a center grant that has an advisory board. If you're on the advisory board, obviously you shouldn't be reviewing it. And then if there's some indirect financial interest that you'll somehow be there, you shouldn't review that. And then the last part, though, says that maybe you can review a few things. That if you have collaborators at an institution, but it's not directly there, you can review if you have indirect financial interest of less than $10,000. If if you freely donate reagents, as long as you give them to everybody, right? So you're not a specific collaborator. Same thing with data. Sometimes there's large research networks. That's okay if you're a member of a large research network. And then the co-author on a non-research publication, I thought that, or a mega multi-authored publication, I thought that was interesting. Occasionally people will write some sort of thing like the status of the field, and there'll be 50 co-authors, right? All the big names will put their name on something saying where they think the field is going to go for energy or for, I don't know, coronavirus research, or something like that. The reality is that can knock off, if you go with the NSF policy, that you can't and those people can review for 5 years because you wrote something with them. Whoa, that could knock out a lot of reviewers. But if you say, well, that was just a commentary, the NIH says it's not important if it's just a commentary, if it wasn't really like a data collection project. So that just gives you some of the ideas. It's not fully, probably defined, but some of the ideas of what conflict of interest is and when people have it. Okay, let's talk a little bit then about compliance. Quite frankly, if we were in class, I would have had Dave Hudson give this talk to you. He is in the Vice President for Research Office, and he is the Senior Associate Vice President for Research and the Research Integrity Office. So he's basically in charge of making sure we all follow the rules for everything. He actually is also in charge of the rules, making sure that we follow the rules to teach you ethics, kind of thing. But if you are not sure about anything related to compliance, you may always email him or call that number. So if there is something going on in your lab that you just can't take, that you think is not right, you can always contact that office. If you need advice about something, again, it's always better to get advice about a conflict or potential problem early rather than kind of waiting on that. The last thing I wanted to talk about were whistleblowers. Let me go back to where I had my whistleblower. I didn't move this slide. I moved it to the end. So the NIH statement says that you should be protected if you're a whistleblower. So what's a whistleblower? Maybe if you haven't heard that term. That's somebody who comes forward to report that somebody is doing something wrong, right? So you say, oh, my advisor is doing scientific misconduct, or my lab mate is doing scientific misconduct. They have a conflict of interest and they're not reporting it. They are misusing animals. They are not protecting human subjects the way they promised to in their protocol. They fabricated data. Any of the things we've talked about, they plagiarized an article. Quite frankly, any of those could go under kind of whistleblowers who are making an allegation of scientific misconduct. ORI here is Office of Research Integrity. So again, normally you actually go to your institution, but what they said is that the institution is supposed to protect you. That when you go, you should be protected from retaliation. So what does that mean for whistleblowers? Here's the NIH kind of whistleblower bill of rights. That whistleblowers are free to disclose lawfully whatever information supports a reasonable belief of research misconduct. Again, you're not necessarily free to go put it on Twitter, though. Like, disclose lawfully means that, again, you should go to the right person in the right institution, somebody above you, not just go tell the world, but if you do it lawfully, you should be supported. The institution should not tolerate or allow retaliation. So you say, well, I can't report my professor, or I can't report a friend of mine, or an older grad student or a postdoc for doing something because I'll get fired, or they'll make my life miserable in the lab. There should not be retaliation against good faith efforts. And then the institutions have a duty to provide fair and objective procedures for examining and complaints. Again, we will have a complaint system here at UVA if there's a dispute or an allegation. And then they have to, have things that are unbiased. We'll talk a little bit about biases next week. And they're going to get, they should get to the bottom of it, find full and objective information about the concerns raised. And they have to handle the cases quickly, right? That's always a difficult one, but, they have to do a thorough review, but they should be quick. And at the conclusions, they have a responsibility to credit promptly those whose allegations are substantiated. So meaning if you are right, they should give you some credit for pointing that out. And so I definitely wanted to make sure that we covered this in this class, because there's probably no other time in your graduate career where you'll be told that you can report something. If in good conscience something is going on that just should not be going on relating to research integrity and research misconduct, you can and should report it. Now, you can always come to me as the department chair, and we can talk about things, as I said, but you can also, if you for some reason felt like that wasn't a good option, go to that VPR's office and Dave Hudson's office, and they are the ones responsible for making sure that research integrity is done correctly here at UVA. Okay, so with that, that's a lot of laws. We'll have a couple case studies this week on these kinds of things, but I hope you see that you need to follow these rules, that you need to be at least knowing that these kinds of things exist.